கேட்கல.
சார் பிபி சார் ஆடியோ அன்மியூட் பண்ணுங்க சார்.
ஓகே அன்மியூட் பண்ணு.
சார் லைவ் வந்துருச்சு. Good morning to one and all. Welcome to day 2, session 1. The chairperson of this event is Dr. P. Pandian, Assistant Professor of Chemistry, Periyar EVR College. And the speaker is Ramakrishnan Sethu from USA. A small announcement for all the participants. Due to academic work of Dr. Sukumar of session 3, we preponed our session 3 to 12:30. That means we are continuously having section 2 and 3. Participants are requested to join the same link. That means you no need to sign in join from session 3 link. Just you have to stay in the session 2 link. We will continue session 3 in the same link. Thank you. Now I hand over the session to the chair.
A very good morning to everyone connected in the second day of 2 days international webinar on. Hello, sir. Is it audible?
Yes, sir. Audible, sir.
A very good morning to everyone connected in the second day of 2 days international webinar on research evolution towards current scenario, which is being organized by our postgraduate and research department of chemistry, Periyar EVR College, Autonomous, Trichy, Tamil Nadu, India. First of all, I thank and congratulate the organizers, Dr. K. Thanigasalam and Dr. C. Pragadishwaran, for arranging this wonderful international webinar. Before introducing the chief guest, I wish to welcome the gathering. I am happy to welcome our college principal, Dr. J. Sugandhi, our resource person, Dr. S. Ramakrishnan, our head of the department, Dr. C. Thangavelu, faculty members of chemistry department and other departments of Periyar EVR College, faculty members, research scholars, and students from various institutions all over the world to this webinar. The resource person of this session in the University of Iillinois Urbana, Champaign, Iillinois, USA. His research area is bioinorganic and medicinal chemistry. He did his UG and PG degrees from Madurai Kamarajar University and PhD from Bharathidasan University, Trichy. He passed the CSIR JR of examination also. He was the first rank holder in BSc chemistry. He served as a postdoctoral research fellow in well-reputed universities and research centers in Japan, France, and in USA. He has published more than 20 research articles in international peer-reviewed journals, which include ACS. Most of the journals have the impact factors of an average of 5. I am proud to mention that one of his research articles was published. His articles were cited in various international journals in 1054 times by various researchers. He has received many best paper awards in international conferences organized in various countries. He conducted a DST-sponsored winter school in bioinorganic chemistry in IIT Mumbai and organized a workshop in Bharathidasan University, Trichy. He served as a college teacher in France in the year 2012 to 2013. His academic background and achievements are going lengthy. I have just highlighted only few things. So we have a right personality as a resource person for this session. We know that one of the Nobel Prizes is given to physiology or medicine. But the Nobel Prizes for chemistry were given to the field of biochemistry or medicinal chemistry for more than 1 or 2 decades, except very few. In this context, he is going to deliver a lecture on Metalloenzymes, novel target for antibiotics. I hope he will entertain us with his scientific speech. On behalf of our chemistry department, once again, I welcome our resource person, Dr. S. Ramakrishnan. Now, I hand over the platform to Dr. S. Ramakrishnan, our resource person. Thank you very much. Have a good day. Welcome, sir.
Thank you very much for your wonderful introductions. So I would like to thank respected principal and head of the Department of Chemistry, and especially I thank Professor K. Tanikachalam for giving me the great opportunities. So with these small introductions, without further delay, I would like to talk about, the novel targets for the antibiotics. So is the metalloproteins. So though it has all the antibiotic has its original target like membrane disruption and DNA damages and stopping by protein synthesis by targeting ribosomes. The metalloenzyme would be the novel target. So once you listen to the lecture, you will know how that metalloenzyme is going to be the target for the, the conventional antibiotics. Okay, with this, I wanted to talk a little bit about the evolution biology. As you know well, 3.8 billion years ago, the world started with anaxic condition. Only the nitrogen was there. So there were no oxygen. Imagine the life was not extended. Life is initiated only because without oxygen, we are not able to live. So only the bacteria and microorganisms were able to live at that anaxic conditions. If you see here, sorry, I wanted to use this. If you see here, the availability of reduced form of, because there is no oxygen, so you could obviously expect that reduced form, no oxidized form. The reduced form of iron and the reduced form of sulfide and other all materials are reduced. So the microorganism took advantage of these materials and the metabolic pathway inherited using this reduced iron and sulfide. So after certain period, because of that, photosynthetic organism called cyanobacteria, that is also one of the microorganism involved in a photosynthesis and produce the oxygen. As you know well, the photosynthesis reactions is used to evolve oxygens. So it's a great thing because it's slowly, the oxygen is slowly, coming and trying to deposit in the environment. But the problem started there. Once the oxygen slowly produced, the problem started there. You could see that the oxygen slowly oxidized iron 2 to iron 3. That's the problem, right? Because the organism using iron 2 plus as one of the metabolic pathways. So it uses, it's a building components in associated with the iron 2 plus and sulfide and whatever in the reduced form. Once oxygen deposited in the atmosphere, all the metal ions is oxidized. Now there is a demand for metal ions in order to perform the biochemical reaction. But that is a different scenario. I don't want to talk about that. It is a big topic. Why, once how this microorganism cope up with the, the metal available other than iron. So that is different topic. But I am going to talk about that problem with the oxygen. Once the oxygen accepts the electron, you see slowly start deposited in the atmosphere, and the oxidized iron formed the ferric hydroxide. You know that as a chemist, you know that ferric hydroxide is insoluble in water. So obviously, as I said earlier, that microorganism no more using iron. So it's trying to use other metals. So now once it accepts the electron, you could see here the diagrammatically I am showing the iron initially it was higher in the geological point of scale. You could see that iron is higher at point of time, means 2.3 billion years ago. So once that oxygen evolved, slowly deposited because of the photosynthetic organism, you could see the oxygen slowly trying to deposit in the atmosphere, whereas dissolved iron went away. Next, now you wanted to know about that how this oxygen getting electrons. You know that this is the molecular orbital, simple molecular orbital diagram of oxygen with the 2 unpaired electrons. You see here 2 unpaired electrons in the antibonding level. So it's also in the other case, you could see the organic biomolecules such as amino acid, nucleic acid, fatty acids, lipids, and so many things. It's organic biomolecules. That is, the antibonding paired electrons. So it is very difficult to take up the electron transfer reactions from, between the organic biomolecules and oxygen. However, the oxygen takes up 1 electron at a time. It's not taking, 2 simultaneous electron or 4 simultaneous electron in such happen in other cases. But here in organic molecules, it has only taken 1 electron at a time, and the reaction also very slow. You could see here. If you one second. So you could see here. If you see here, the evidently it is showing that the oxygen takes up 1 electron to become a superoxide. If you see the redox potential of this reaction is negative, that process is very slow. That's what the organic biomolecules in the aerobic condition is not degraded. So now this is superoxide, it's a lifetime, and the half-life period of this superoxide is very, very slow. Once the superoxide form, then it's immediately dissociated or dismuted into, here you see, dismutated into hydrogen peroxide. So you could see that redox potential is positive. It's a quick reaction. Once the hydrogen peroxide is formed, then if there is any electron donor, because it's a hydrogen peroxide is a strong oxidant than superoxide as well as the oxygen, then once there is any electron donor, it's readily accept and produce the deleterious radical. You know that this hydroxyl radical is really pretty dangerous. So if the radical is a nearby biomolecule, it's easily accept the hydrogen and leaving as a damage, like it damage protein, it damage lipids, it damage DNA kind of stuff. Eventually it lead to cause a disease, some oxidative, some neurodegenerative disease. And finally, that it is becomes water. This electron transfer reaction, this oxidation is a sequential, the electron one by one by one by one. So it's not, at a time, it's not taking place 4 electron and 4 proton electron transfer reaction. It's sequentially. So here, I want to talk about these oxygen and hydrogen peroxide and superoxide. So if you see oxygen and hydrogen peroxide, both are non-polar, whereas the superoxide is polar. Since hydrogen peroxide and oxygen are polar, it can easily penetrate into the cellular membrane. So maybe it exchange, back and forth. It get into the cells and coming out of the cells. So get into the cells and coming, but the superoxide is negatively charged. So it's not able to penetrate because the lipid membrane is, consists of fatty acid, fatty acid and the lipids are negative charged in the negative repulsion. The superoxide is not able to get into the cytoplasm, but oxygen and the hydrogen peroxide are able to get into the cytoplasm. Once the hydrogen peroxide get into the cytoplasm or oxygen get into the cytoplasm, then obvious questions. So how it forms, like what is the sources of the hydrogen peroxide? What is the sources of the hydrogen peroxide? Where it forms? Once the oxygen get into the cytoplasm, you could see there are 2 different sources. One is endogenous. Endogenous means within the cells. The other one is exogenous. It's outside of the cell. The inside of the cell, once oxygen get into the cytoplasm, it collide with the flavin containing enzymes. So the flavin containing enzyme, if you see here, this is the flavin moiety. There are certain enzyme it uses in the electron transport chain, particularly, which is like fumarate reductase and succinate dehydrogenase and NADH dehydrogenase. Those enzyme uses these electrons. When the oxygen collide with the flavin, so the electrode potential, you could see that it's a slow reaction, but it still happen. So once, if you see here, this is also negative and this is also negative, the electron potential matches and it steals the electron, and you could see that it's eventually it forms the hydrogen peroxide as well as superoxide. Once the superoxide formed, as I said earlier, its lifetime is very small. Yeah, is obviously you could see the accumulations of hydrogen peroxide inside the cell. The other one possibility, that's like the reduced free ion. As I said in the evolution biology, the oxygen steal electrons, the free ion of iron 2+ and becomes, superoxide. Once the superoxide form, it's eventually end up in the hydrogen peroxide. So these two are the sources endogenous. Endogenous means inside the cell. So the next question, what is exogenous? What is outside of the cell? For example, this is ROS, means reactive oxygen species. So the reactive oxygen species includes superoxide and hydrogen peroxide. So these are very reactive. So reactive species means very reactive. So as I said earlier, the reduced metal ion and thiols, as said in the evolution biology, it's also available in the environmental too. So if from there you could see reactive oxygen species, and another one is a redox cycling compound. Here it's interesting. What is that redox cycling compounds? So if you see in the bond, there is the leaves falling down in the bond, then it will turn into the yellow color and eventually turn into the purple color. The color is because of it excreting some redox cycling compounds. Once it's excrete the bacteria inside the band, it's directly penetrate into the cell, and where the oxygen is there is, eventually it forms the superoxide and hydrogen peroxide. And another one is lactic acid bacteria. Is a fermenting bacteria. Wherever you use some fermentation process, this bacteria uses. This bacteria uses a hydrogen peroxide as a weapon to kill the counterpart, other bacteria. Only lactic acid only grow on that fermentative process. But if there is any other bacteria is growing, then it will eventually kill that bacteria. The final thing is that it's interesting that, it's immune cells, like I would say, like macrophages and neutrophils, use as a hydrogen peroxide as a weapon to kill the pathogenic bacteria. So for example, once the pathogenic bacteria is inside the body, the first thing these macrophages and neutrophil engulf the bacteria and kill that bacteria. So you could see that there is a duck of war, because the bacteria try to accommodate, try to adapt that environment. But these macrophages using hydrogen peroxide, that reactive oxygen species, as a weapon to kill the bacteria. So these are the exogenous, it's outside of the bacteria. So obvious question. Okay, sources and how. So is there any way, like if that hydrogen peroxide or superoxide enter the cells, it's a deleterious effect. It will kill the cells. But how that the cell cope up with this deleterious species? So you could see the cell is cleverly using defense mechanisms. All of you know, because even though we are chemists, we know that these defense enzyme, like as is a superoxide dismutase. The self dismutation, the rate of the reaction is, the rate constant of reaction is 10⁶ per molar per second. But this is 10⁻10 per molar per second, the rate constant of the superoxide dismutase. So I am going to talk about the model bacterium E. coli. In E. coli, it has 3 different superoxide dismutase. One is iron superoxide dismutase and manganese superoxide dismutase, which is in the cytoplasm, but in the copper zinc SOD in the periplasm. Because in daily process, you could see the hydrogen peroxide as the superoxide, eventually get into the cells, means it's produced in the cells, and this superoxide, and other one is catalases. Catalases degrade the hydrogen peroxide. In E. coli, it has 2 catalases. One is CatG and CatU. And also it has some peroxidases, like it has several peroxidases. So these defense enzymes are the safeguard, are the safeguard for that deleterious effects such as hydrogen peroxide and superoxide. If you see that these 2 uses the hydrogen peroxide. The peroxidases and catalases, the name itself peroxidases. In the catalases, if you see that the catalysis uses two hydrogen peroxide, use the two hydrogen peroxide, and it degrades all these two hydrogen peroxide at a single time. But these one, the hydrogen peroxide, only one hydrogen peroxide. This is more effective. This is more effective than this. So, but the bacteria uses this kind of defense enzyme in order to degrade, in order to defend with the deleterious the reactive oxygen species. So then the obvious question, like, so for example, here I wanted to mention one point. I wanted to emphasize the rate of productions of reactive oxygen species and the rate of degradations of reactive oxygen species are exist in equilibrium. So always these defense enzymes keep that reactive oxygen species on the basal level, so no harm for the cells. But if there is any problems, if there is any imbalance between the production as well as the degradations of hydrogen peroxide, there is a problem arises. Means like the cell eventually going to die. So then how its cells is going to cope up with that? So for example, if there is any defective in enzymes or the overproductions of the reactive oxygen species, eventually it will kill the bacteria. So what we did, we wanted to measure the hydrogen peroxide inside the cell. It's very, very clever method. It is. So what we did, we deleted the scavenging enzyme, hydrogen peroxide scavenging enzyme. For example, if I say, so in bacteria, there is a wild type as well as mutant. The wild type means the bacteria as such, without any modification. And here the mutant means like, we did some modification. The modification such as we deleted the strain. So what is a HPX mutant? HPX mutant means, mutant strain means, so we deleted the CAG. You could see here, the CAG and CAG, two catalases and one peroxidases. These are the main gene that, responsible for degrading hydrogen peroxide. If you delete these, then hydrogen peroxide is automatically, get into the cell. We can measure how much. So what we did, so we set up some experiment. I will go through some experiment. Is everyone knows it's a basic experiment. You could see here. So we set the, one second. So experimental setup. As you know, well, if you want to study about the bacteria, you need to use some plates, some the LB or minimal plate, whatever it is based on the condition what you want. We stick the bacteria on this plate, and leave it for overnight at 37 degrees. If you see here, there are a lot of single colonies. So what you will do for overnight growth, so we will take one single colony and inoculate on some medium. The medium might be different medium. Again, it depends upon what you want. So if you want to study the physiology, the things are different. If you want whatever you want, you can use the different medium based on. But here we use the LB medium, LB Lubria Britannia medium. So once it's inoculate, if you see after 12 hours, it will be like a derbit. So which means its bacteria is already grown, and you can measure how much is the bacteria is grown using simple UV, visible spectroscopy, OD600. So once you see that, then what you need to do other day, you need to inoculate for the pre-culture. What is pre-culture means? Like it's intense. It's for example, the OD might be 4.3. So you need to dilute all the way down to 0.005, and you can leave it for the growth because you need a exponential phase. So as bacteriologists or other people know what is exponential phase, because here all the physiology is very pressed. You can study the physiology. So once you grow to reach until 0.2 OD, then what you need to do, you need to centrifuge and wash the pellet, whatever the medium you wanted to do the experiments. So for example, I did that, and you see the color of the medium is changed because I used the puffer. Here I used the clay puffer in order to measure the hydrogen peroxide. So I resuspended and exposed to air. So once you expose to air, now imagine the wild type has the catalases, but the mutant, HPX minus mutant, doesn't have any catalase. Obviously, we could expect the hydrogen peroxide accumulated in the cells. So what I will do, I will take 1 ml of each periodically. then I will centrifuge, and the supernatant will be used for measuring the hydrogen peroxide. Here I am going to say that, the hydrogen peroxide can be measured using simple method, like, all of you know that horseradish peroxidase and amplexator assays. It's a easy assays. You can do that. If you see that the horseradish peroxidase enzyme uses iron 3, and it reacts with the hydrogen peroxide. So for example, wild type doesn't produce the hydrogen peroxide, but mutant can produce the hydrogen peroxide. Once the hydrogen peroxide is formed, the enzyme is oxidized itself into the higher valent oxo species. Then it abstracts the electron from the amplex rate. This is EIR, means amplex rate, that is the substrate, by sequentially. You could see that compound 1 and compound 2, and finally you could see that the oxidized version of the risopyrene. The amplex rate is oxidized into risorufin, and it will be measured the fluorescence. So once you measure the fluorescence of oxidized risorufin, because it's directly proportional to the concentrations of hydrogen peroxide, and if you know the standard concentrations of hydrogen peroxide, already known volume of concentration of hydrogen peroxide, you are able to see that how much hydrogen peroxide is produced. Here you go. So if you see the wild type, there is no hydrogen peroxide. I've taken the sample in 10 minutes, 20 minutes, and 30 minutes. You see, like, there is no hydrogen peroxide is produced. But in the HPX minus, because it doesn't have, again, and insisting, it doesn't have any type of catalase or peroxidases in order to degrade the hydrogen peroxide. You could see here, the concentration of, hydrogen peroxide is steadily increasing. And the rate of the hydrogen peroxide is, the concentrations of hydrogen peroxide, and, it's 15, rate of the hydrogen peroxide is 10 to 15 micromolar per second. For example, this one is normoxia. The normoxia condition is 20% of oxygen, only 20%. So if you want to measure the higher concentration of oxygen, you could see that, the saturated oxygen, you could see that it's steeply increasing. Even it's unmeasurable. Even you don't know how much hydrogen peroxide is formed, because you could see the difference between 20 to 100%. So here, we have a device in order to measure the hydrogen peroxide. Now, once the hydrogen peroxide enter into the cell, so if we don't have any defense enzyme, now once the hydrogen peroxide goes inside the cells, we could see what are the things are happening. So first, once it's entered into the cell, there is a sensor, because hydrogen peroxide is there, and the cell is cleverly have some setup in order to, degrade some unwanted things. So as here, the E. coli is a gram-negative organism, and you see here the OxyR, that is the transcriptional activator, and OxyR uses is a dithiol group in order to sense the hydrogen peroxide. Once it senses the hydrogen peroxide, you could see that the sulfonic acid, the one thiol group reacts with the hydrogen peroxide. You see that it oxidized into sulfonic acid. So you could see that it reacts with the hydrogen peroxide, take hydroxyl group, and so that reaction means it take S minus, HO plus, and it becomes a sulfonic acid. Then it forms, so it will further oxidize into disulfide. You see that, self-condensation, and it becomes a disulfide. This is reduced oxyar, this is oxidized oxyar. So you could see that conformation. So I didn't show the slides, like you could see the conformations of oxidized versus reduced versus oxidized. Then once it's oxidized, it's suitable for, binding with recognizing with the RNA polymerase. Then it will eventually induce the gene which is responsible for the hydrogen peroxide sensing. So those, now it's, it will induce the enzyme that is, ready for degrading hydrogen peroxide. So likewise, in gram-positive, there is one transcriptional regulator, is also the hydrogen peroxide sensor. Its name is PERA. You see, the PERA is a de-repressor. Here, the mechanism is entirely different. For example, under normal condition, is a positive organism, the E. coli is the negative organism, gram-negative organism. If you see that, the PERA, the transcription is bind on the promoter regions of the in genes, the genes that wanted to, react against hydrogen peroxide. So under normal conditions, it binds on the promoter regions and it doesn't allow the RNA polymerase to synthesize the gene. But if there is any hydrogen peroxide, you see the hydrogen peroxide is there, it oxidizes the protein, leaving RNA polymerase to synthesis the inducible gene, which are CAD-A, that is different genes, CAD-A and AHBC, MERK-A, that is, everything is responsible to degrade the hydrogen peroxide or whatever happened inside the cell, to keep the cells alive. So then I wanted to talk about the PERAR protein. The PERAR is a homodimer protein. You could see here, it is a dimetal, means like this zinc in one side and iron or manganese. I didn't put the manganese in the other side. This is, the zinc is very important for keep the structure formation, and the iron is to, that iron is readily sense the hydrogen peroxide. So we will talk about a little bit later. So, and we took the structures, and structure, you see this apoprotein. Apo means without metal. You could see zinc is for only the structure, but the regulatory side is no. So it's like open structure. This is like open structure. When the metal forms, you could see the caliper-like conformation. The conformation is changed. As I said, in oxyar, even the PERAR, you could see that conformation once the metal is bound. This conformation is very suitable for binding on the promoter region. So if you zoom the active site, you could see that it's a square planar compound and it's a 5 coordination and 2 histidine and 2 aspartate in the square planar in the equatorial plane, and H93, histidine 93 in the axial plane, and there is one vacant phase that is readily available to sense the hydrogen peroxide. So here that hydrogen 37, this is very important in order to have the caliper-like conformation so that it's able to bind on the gene. So I'm quickly, so there are 2 forms of PERAR. One is iron PERAR and manganese PERAR. Manganese PERAR is insensitive to hydrogen peroxide, but iron PERAR is a metalloprotein, metalloenzyme. So it's sensitive to hydrogen peroxide. And it's following the mechanism is a Fenton mechanism. You are familiar with the Fenton mechanism. Iron reacts with the hydrogen peroxide and releases the hydroxyl radical. Once the hydroxyl radical is formed, the lifetime is very, very small, pretty smaller than the superoxide. Then immediately, grab the one hydrogen from, nearby biomolecules. So we see here the histidine oxidized in the 2-oxohistidine, which is the histidine coordinated, the H37 coordinated into the iron site. Once the hydrogen peroxide comes, then it produces the hydroxyl radical or ferryle intermediate, and to take the electron away from the coordinated histidine. So we use the MALDI-TOF mass spectroscopy in order to analyze the data. So I don't want to get into detail. So if you talk about the gene regulations back in E. coli, oxyar, they are regulating is 200 genes, but I don't want to talk about that 200 genes because still there are a lot of genes to be analyzed. What is the functions? So I am taking particular genes which is responsible for DNA repair enzyme. Once the DNA is damaged, how the DNA repair enzyme repair the DNA damage. And also, as once the hydrogen peroxide entered into the cell, how this, degrading the hydrogen peroxide. And third one is iron sulfur cluster that I'm going to talk little, in a minute. Then, some metal homeostasis. The MNTH is the manganese importer, and FAR is regulating like, iron homeostasis, and DPS is a DNA binding protein and the starvation. So I'm going to talk about a little bit this stuff. So since I am talking, once the hydrogen peroxide enter into the cell, and it will target different. One is DNA. The first one is DNA. We wanted to study the DNA. So we always compare the HPX minus cells. And so HPX minus cell, you remember always the hydrogen peroxide is accumulated, whereas the wild type, it has the enzymes to degrade the hydrogen peroxide. You could see that, so here, I am showing the morphological figures. Under the microscope, we see the live bacteria. So the fermentation, DNA fermentation is the hallmark of DNA damage. So if you see the wild type, there is no damage. You could see, the bacteria is moving. So I don't have the the video, but you could see in video the bacteria, you can see in naked eye the bacteria is moving. But here, since the DNA damaged, you could see the fermentation, cell septation, we should say that is cell arrest, and you could see it will ferment, no more growth, no more further growth. So, and the order of fermentation, you see here, now I talked about the repair enzyme. If you delete the repair enzyme, you know, and the background of HPX minus, you could see the higher DNA fermentation. So the order is wild type, there is no fermentation, HPX minus little bit, but in HPX RECA minus, because here the RECA is there. RECA, you know, will take care of to fix that whatever DNA damage. But here, the RECA is not there, so you could see the DNA damage, eventually the cell is going to die. The next one is like then damage, then now we wanted to know like whether it's because of iron or because of hydrogen peroxide or because of what it's the DNA damage occurs. So if you see, first I wanted to, the first one, you know that because wild type on a hydrogen peroxide, the hydrogen HPX minus, the hydrogen peroxide accumulated in the hydrogen in the HPX minus chain and damage the DNA. Now we wanted to see like the hydrogen peroxide reacting with other molecules, such as metals. So I wanted to see whether the iron is responsible for the reaction with the hydrogen peroxide and produce the hydroxyl radical that damage the DNA. You see here, if you see, this is the plating assay. I would say that is the cell survival assays most of the people might have done in collaborating with the microbiology people. If you see here also I am using wild type as well as the HPX minus cell. If you see here, these closed circle is the wild type. You see when I treat with the metal chelators. So for example, if you add diaphragmine or dipridal, you could see chelate the metal. It cut into the cell and chelate the metal. It is not leave the metal ion to react with the hydrogen peroxide. If you see here, so this is the wild type. There is not much changes, you know, before and after diaphragmine. So if you see here, you know, the HPX minus cell, you know, the cell survival is decreasing and slowly, you know, recovering. But when you add the diaphragmine, you could see that it's pretty good. It's recovering some cell survival. So this shows that, you know, the iron also involved. The dipridal chelates iron that helps recovering the cell survival. So first we saw that it's a hydrogen peroxide. And now we know that it's iron is involved for the DNA damage. Then the obvious reaction is the Fenton reaction. So iron reacts with the hydrogen peroxide and produce the hydroxyl radical. So we have a method to detect the hydrogen peroxide. You can trap the hydroxyl radical, and we use the tempo method, you know, and using EPR, we try to detect the hydroxyl radical. You see wild type, there is no the characteristic EPR spectrum for that tempo radical. So if you see the HPX minus, you know that the hydrogen peroxide is there, then it eventually, you know, it reacts with the, you know, an iron, then you could see these three characteristics for tempo radical. So if you treat with the catalase will degrade, means HPX minus, that if you add catalase on top of it, then it will degrade. You could see, you could see there is, you know, signal is gone. The next one, if you treat with the chelator, such as dipridal or diaphragmine, that is also no signal. For example, if you treat with 2 micromolar, you are enhancing the concentration of hydrogen peroxide. You could see that increment of that signal. And even when I add 10 micromolar hydrogen peroxide on top of the HPX minus chain, you could see the large the indications of, like, you know, hydroxyl radical also involved. So it seems like, you know, the Fenton reaction is the major reaction in order to damage the DNA. So now we could claim, based on these results, as you know that once the oxygen is coming from, you know, it becomes superoxide and hydrogen peroxide, whatever the ion source available, as you know, well, it damages the DNA. It's a chemistry people will know these reactions. It's a familiar reactions. So now we can claim that based on that result, we can claim that it's you know, the DNA is the target for the hydrogen peroxide. The next thing is that, you know, obvious question, we know that the level of hydrogen peroxide and what is the level of iron inside the cell. So we measured the iron inside the cell. You could see the difference between using the EPR, you know, that it's iron 3 active. Then we can measure the whole cell. You know, we have a setup to measure the, you know, iron level inside the cell. So you could see that difference, wild type and HPX minus at least 3-fold higher in the HPX minus. The next one, where does the iron come from? That is the question, right? So once the hydrogen peroxide accumulated, where is the iron come from? So it's a big, topic to cover that, but I am not able to cover that. So I will take it very, very sharply into, like, we tried the iron. Obvious thought is the iron supposed to from iron storage protein. The E. coli having 3 different iron storage protein. One is bacterioperitoin, another one is ferritin, and third one is DPS. So we tried to measure the iron in the background of HPX minus. So we didn't see anything involved in the source of iron. But we are able to find out, the fumarase, agonitase, and which is involved in the DCA cycle, the 4-iron 4-sulfur cluster enzymes, involved in the metabolic pathway will be the source of the hydrogen peroxide. So will be source of the iron. So the fumarase and agonitase is a 4-iron 4-sulfur cluster, which is involved in TCA cycle, and the EDD enzyme involved in the alternative pathway of the DCA cycle, which is eternal derivative fermentic pathway. The IPMA and the DHDE also used in the branched chain amino acid synthesis pathway. So yeah, these are the enzyme 4-iron 4-sulfur cluster will be the source of iron. So we measured that. Obviously, like if you has iron, the hydrogen peroxide will target the iron sulfur cluster, and you could see that when there is no hydrogen peroxide, it's 100% active. When you increase the hydrogen peroxide, it's eventually decrease the activity. So IPMA activity at least inactivated by at least at least 95% inactivated within 5 minutes, using 6 micromolar in vivo. So the cell extract prepared in vivo. So then we wanted to see that is in vitro, this is in vivo. In vivo means, like, we grow the cells, then add the hydrogen peroxide inside the cell, then we prepare the hydrogen peroxide. Then we measure the hydrogen peroxide, just without hydrogen peroxide, you could see the percentage of activity 100%. When I add the 2 micromolar hydrogen peroxide and expose for 5 minutes, you could see most of the activity is gone. So then we wanted to reactivate. So when reactivate, we need iron and TTT. If you add iron and TTT, you could reactivate only 60%, which means, still there is some there is some form which are not able to reactivate. So we have some method to reactivate, like the complete cluster. Here, if we add iron TTT, the case is a protein, and another cysteine. If you feed all these things, then you can build the cluster. So and also we measured the purified protein. We also measured the hydrogen peroxide, means like the iron level in UPR as well, as I told already. You could see without the hydrogen peroxide, there is no iron. There is a slide, because slide in the wild type, as you know, that there is a 60 micromolar iron, you could see. But when we treat with the hydrogen peroxide, and you could see that a huge amount of iron is available in the cellular environment. So the hydrogen peroxide, so based on the result, we can conclude that the hydrogen peroxide leaches out the iron from IPMI. Means IPMI means it's iron sulfur cluster, which is involved in branched chain amino acid pathway. That is, leucine, isoleucine, and valine. So that one is like, it's a mechanism how this iron sulfur cluster, binds the substrate and leave the product in the metabolic pathway, and how it's damaged by hydrogen peroxide. If you see hydrogen peroxide attack these enzymes, and it forms like a higher valent oxide species, later on it becomes loses the iron 3+, and OH minus is a pentan reaction is going on here, and you see that 4 iron sulfur cluster changed into 3 iron sulfur cluster. So that is what, the 60% of 3 iron sulfur cluster was reactivated by electron donor. That is the DTT. I have shown the previous figures is a DTT and iron 2+, to rebuild the iron sulfur cluster. If there is, so this lifetime is also, it's kind of small. So eventually it will, loses all iron sulfur cluster, becomes apoprotein, and you need all these materials to build the cluster and put on the apoprotein and reactivate it. That's what I shown in the figure I wanted to show in front. You see, if you see here, iron TTT is a 3 iron 4 sulfur cluster, and this one is a 4 iron, means this one is completely, apoprotein is built by the iron sulfur cluster and incorporated into apoprotein to see the complete activity. So based on the data, the previously I showed, this is the metabolic pathway. You could see that when wild type, when I knock out that EDD, which is involved in the Edner-Dewdorf pathway. So when you knock out, you could see the difference. It's there is a growth defect. It's also, you could see here is a wild type, and when I treat with the hydrogen peroxide on HPX minus, is completely, completely stop growing. When I add the product means like, these enzyme involved in synthesizing this product is isoleucine, leucine, and valine. You could see the recovery of growth. So if the hydrogen peroxide damage the iron sulfur cluster which is involved in the pathway, then you can't see any isoleucine, leucine, the branched chain amino acid. Eventually you see the plot on the curve. So when you throw the product, you could resume the growth back. So this is the pathway. I wanted to show that this is the pathway for leucine, isoleucine, and valine is IPM and DHDA is involved in this biosynthesis pathway. So based on these data, we can say that initially I claimed DNA as a target for a hydrogen peroxide. Now the iron sulfur cluster also the target for the hydrogen peroxide. The third, is there any other? Because iron sulfur cluster means not only iron sulfur cluster, you could see mononuclear protein is a heme containing group as a SAM dependent iron sulfur cluster. So we are trying to search, is there any other metallo target for the hydrogen peroxide? So are there any other family for the hydrogen peroxide? One is mononuclear, dinuclear, and SAM dependent and heme containing. So we wanted to proceed with the mononuclear iron protein first. So we characterized in our lab, we characterized 5 different iron protein. One is threonine dehydrogenase, cysteine deaminase, DAHP synthase, ribulose 5-phosphate 3-epimerase, and peptide deformylase. These are involved in various biosynthetic pathway. For example, I know I'm not going to concentrate all these 5 because we have done all these 5 experiments, 5 enzymes. We characterize 5 enzymes, but I wanted to talk about 2. So one is ribulose 5-phosphate 3-epimerase, which is involved in pentose phosphate pathway. So it's an alternative pathway for the TCA cycle. If the TCA cycle is not working, then it will uses the alternative pathway, that is the pentose phosphate pathway. The another one is peptide deformylase. It's very, very important, essential gene, because once the protein is synthesized, it's always synthesized with the formyl group. So formyl group is very toxic to the cells. So actually what this peptide formylase is doing is deformylate. So these 2 enzymes, I have taken, in order to present this in detail. So ribulose 5-phosphate 3-epimerase. So, the short form is RPE. So what actually doing these changes ribulose 5-phosphate to cellulose 5-phosphate. That is, both are ebimers and the intermediate. So what actually this enzyme is doing, like, the pathway having a lot of enzymes is gluconate dehydrogenase. Okay, this is ribulose phosphate isomerase. This is RPE. So you see that from here to here, like ribulose phosphate to cellulose 5-phosphate. So we characterize all the enzymes. So all the enzyme and wanted to see why that biosynthetic defect is happening. So then, what actually this RPE is doing, that's epimerase. Epimerase, the changes in the carbon atom, if you see here, is above the plane, and if you see that the hydrogen is below the plane. So this is acid-base catalysis, and this enzyme, the active site, is stabilizing the intermediate. You see here the carboxylate ion of the substrate, the enzyme is stabilizing. It's especially used for the stabilizing the intermediate. This is taking one proton and putting here and become a stereo interconversion from ribulose to cellulose 5-phosphate. So, I want, this is the method that I use. And especially I wanted to tell you here, I added EDTA when I washed the cell. The reason why I added EDTA, because I'm going to work on the metalloprotein. So you should not exchange the metal, because always the metalloprotein, the small, is a thermodynamic equilibrium. It's a thermodynamic equilibrium, go and come off. So when you wash the cells, it's taking the metal, like the extracellular metal, so that you can see what is inside the enzyme, what is the metal available inside the enzyme. So this is the enzyme assay, and here, this is it's a complicated assay. So it's an enzyme coupled assay. So finally we measure the NADH degradation in the in UV visible spectroscopy. So at 340 nanometer. So we analyzed all the enzyme involved in the pentose phosphate pathway, and we tested with the hydrogen peroxide, sensitivity of the hydrogen peroxide. You could see that the enzyme RPE, ribulose biphosphate 3 epimerase, is sensitive to hydrogen peroxide. None of the other enzyme for RPI, TKT, transcriptase, aldolase, or 1 dehydrogenase is not sensitive to hydrogen peroxide. Only RPE is sensitive to hydrogen peroxide, which shows that the RPE has the ion in the active site. Then I take the RPE and reacts with the hydrogen peroxide and try to reactivate. So when I reactivate, only 80% I am able to reactivate, and remaining 20% I am not able to reactivate when I add the metal back after treating with the hydrogen peroxide. It shows that only the hydrogen peroxide oxidize the iron 2 to iron 3. The iron 3 is not binds to the apoprotein. So when you add iron back, it shows some certain level of activity. So it shows only 80% activity. So 20% we suspect is a polypeptide oxidation, which is not able to activate. So if you see the compare with the manganese, is more or less equally active, and zinc is less active. So zinc is very less limited activity. So we also purify the protein to make a apoprotein. As chemistry, we do like we can make a complex with the same ligand. Likewise, you have to consider the apoprotein like a ligand. You can vary the metal. So if you see here, I made a apoprotein and treated with the iron and manganese, cobalt, and zinc to see whether it has the sensitivity to the hydrogen peroxide. Only iron enzyme has the sensitivity to the hydrogen peroxide, none of the other metal. We have tested several metals, so I am taking this is for the series, but iron containing enzyme is sensitive to hydrogen peroxide. So initially it was shown that, in the literature shown that it's zinc containing enzymes. So when you purify the protein, we are purifying protein in an aerobic environment. The iron enzyme easily, even though if you have iron enzyme, it easily reacts with iron slowly during the purification. And if you see the Irwin-Williams series, zinc is a strong binding affinity towards the metals. So you see here, you can keep the apoprotein and be characterized like RPE is originally using iron in vivo and in vitro. So now then the peptide deformulase, as I said, that it will deformulate, toxic deformulation into amino group. And you could see here, wild type shows 100% activity, and HPX minus. HPX means it accumulates hydrogen peroxide. You see that activity is decreased by 18%. If I knock out the knockout on the background of HPX minus, this is manganese importer, because manganese also reactivate. Manganese also reactivate. We are doing in vivo. It's MNTH is importing manganese and putting on the place of the apo enzyme. So when you knock out the manganese importer, it is not the further it decrease. The third one, when you add the manganese, so the manganese, so then it's reactivating. So the manganese is helping under the oxidative condition. If you see here, so is the iron enzyme, when hydrogen peroxide is inactivated, then RPE is oxidized. So at the time is iron is falling down, then this oxy are importing manganese, and manganese come in, and manganese is used as a placeholder during oxidative stress. So this is, now I am claiming that non-redox iron enzyme also target for the hydrogen peroxide. So first is DNA and iron sulfur cluster. That one is mononuclear iron protein. So now we are under process of the SAM dependent and heme dependent. So all these enzymes we wanted to test whether it's a target for the hydrogen peroxide. So the process is going on. So the overall picture, I wanted to show that, the hydrogen peroxide is produced in the various sources, like as I said earlier. And once it's formed, it reacts with unincorporated or incorporated ion and produce the hydroxyl radical. The hydroxyl radical damage the DNA. That is the major event. So iron sulfur cluster, we have seen that is also target for the iron sulfur cluster and leaching the iron out. And also mononuclear ion, that is also leaching the iron out and produce the hydroxyl radical.
Ramakrishnan sir. You have 5 more minutes, sir.
Yes. Yeah, I can finish it. 5 more minutes. It's about to finish. Okay. So these are the these are the targets for the hydroxyl radical. So once this damage is happen, there are certain machinery inside the cell. For example, DNA damage is repaired by DNA repair. DPS, this kind of sub machinery is repair the iron sulfur cluster damage, and manganese is a placeholder if the iron enzyme is oxidized. So the cell has the clever strategy. Even though there is a repair damage, the cell has the clever strategy in order to repair. That's what every day it's going. Our cell is damaging and fixing, damaging and fixing. So we don't know that. So since we have the device in order to study all these things, we are able to see, hey, this is what is going to happen. This is our, this is happening in day-to-day life. Day-to-day life, you could know that, million cells are dying and million cells are repaired. So this is what is going. So now this is the overall picture, in the oxidative stress. So now the final questions are the Metalloenzyme is the target for antibiotics. So it's very interesting. Now we are working on that. The stuff is like, in the recently there was a cell paper they produced the common mechanism for cell death induced by bactericidal antibiotics. There is different kind of active antibiotics. One is bactericidal, which means cell death, and bacterial static, that means stop the growth and resume. So now you could see that they used conventional antibiotics, some aminoglycosides, quinolone and beta-lactam. Then it's get into the cell and they are seeing different metabolic change, depletions of NADH. And if there is any depletions of NADH, you can see that superoxide is formed. They measure the superoxide. So once the superoxide is formed, eventually it forms the hydrogen peroxide. So you could see here the iron sulfur cluster. So I thought that hydrogen peroxide is the target for iron sulfur cluster. So they said that it's targeting iron sulfur cluster, and it loses. So finally, it's a hydroxyl radical that I have talked about that. That hydroxyl radical damage the DNA and eventually death. Now what we are trying, so okay, whether it's producing ROS or not. So it's directly target the metalloprotein. You see that iron sulfur cluster also metalloprotein. So now we are curious. We have some data, but because of the time limit, I wanted to conclude these lectures. So finally, I acknowledge my group, like my senior professor, Dr. J. Imlay, and I also acknowledge my colleagues and my 2 or 3 of students working under me. I also acknowledge. I thank all of them for contributing those research. And I thank you very much, Pondian sir, and principal and head of the department, Periyar UVR College. And thanks for the participants. And if you have any questions, please don't hesitate. You can ask. We can go ahead. Now I am hand over the session to organizing committee. Thank you very much.
Thank you very much, sir, for your nice and wonderful presentation. Actually, people much interested in inorganic chemistry find a little bit of hesitation on bioinorganic chemistry on that area. But you have explained everything in a simple manner, in a crystal clear manner, the bioinorganic chemistry. So the participants will be actually interested in studying bioinorganic chemistry after listening your speech. Thank you for your enlightening speech, sir. No, only one or couple of questions are there. A participant, Kanmani, asked a simple question: reactive oxygen and a nascent oxygen, both are same or not? This is the question asked by a participant. Reactive oxygen and a nascent oxygen, whether both are same or not?
I would say that, as far as my knowledge concerned, I have also heard about the nascent oxygen. That's a good question. Even, I am working on the oxidative field for 7 years. I never thought about that nascent oxygen. It's a wonderful question. But obviously, I'm telling that it's not reactive oxygen species, because we know that what is reactive oxygen species. The nascent oxygen, even I don't know how does it form inside the cells. I never heard about. Thank you very much. Maybe I can have a look whether that nascent oxygen is a wonderful question. I appreciate it.
Another question from Jacob Mahadevan. Are you saying that salt is essential for our cell activation and decomposition.
Cell, yes, obviously, it's very important. It's very important. So otherwise, you could see that. Actually, I didn't talk about the effect of superoxide. Actually, we recently published one PNAS paper. Actually, the anaerobic bacteria happily grows in anaerobic environment. If I take to aerobic environment, it stopped growing. So and we figured it out. If you knock out the superoxide dismutase, the cell doesn't grow. If I put back superoxide dismutase, if we can supplement, we have the genetics, we have the genetic tools to express the superoxide dismutase, and it will happily grow in the presence of oxygen. So obviously, I really appreciate that question. That question is really wonderful question, because superoxide dismutase, because that is the one is finally facing the first facing the reactive oxygen species, the very important. If you knock off the superoxide, you could see that it's not going to work out. So superoxide enzyme is very essential.
Thank you very much, sir.
Thank you very much.
Thank you for your nice explanation and interaction. Now,
thank you very much, sir.
Thank you. On behalf of the chemistry department, or Periyar EVR College, I wholeheartedly thank the resource person for his wonderful speech in our international webinar. Sir, once again, thank you very much, sir. Thank you, Ramakrishnan, sir.
Appreciate Periyar EVR College team. During this situation, you wanted to feed some, the scientific thirst to the, scientific community. I really appreciate once again Professor K. Thanikachalam and your team, chemistry team, having some nice effort. So I really appreciate it. Have a nice session again.
Thank you, sir. Thank you very much, sir.
Bye.
Here is an important announcement for the participants. Please note, due to the speaker's unavoidable emergency academic work, the third session of the day 2 has been preponed to 12:30 instead of 2 o'clock. See, actually, the session 3 of day 2 will be preponed to 12:30. Participants are requested to stay in the same link day 2, session 2. See, you need not go to the third session's link. Please note, you can continue in the second session's link itself. You need not go for the third session link. Okay. Thank you very much. Thank you, participants. Thank you very much. Thank you, organizers. Thank you, sir. Thank you. Thank you, one and all.
Thank you very much.
Thank you, Pandian, sir. Thank you, Ramakrishnan, for your nice presentation.
Thank you, sir.
It is really wonderful. I think most of the students and participants are enjoying your lecture. Thank you, everyone. We will meet in the.
Yeah.
Tell me, sir.
So you can disconnect, right? Or can I disconnect?
Yes. Yes. We can do it. No problem. Thank you for the participants and everyone. And shortly 11:30. 11:30 we will join the next session. Thank you.
You can use, participants, you can use the second session link for the next session.
Yes.
For the 11:30. Okay. For both the second session and third session, you can use the second session's link itself. Thank you, participants.
Thank you, sir.
Thank you. Thank you.